Hi, welcome back to video 3. In this third video, we're going to look at differences between prokaryotic and eukaryotic transcription, as well as the types of processing needed in eukaryotes before messenger RNA is formed. So transcription in prokaryotes and eukaryotes is very similar, but the main difference is that because in eukaryotes, we have a membrane-bound nucleus, this means that transcription will occur in the nucleus where we read our DNA and we make our messenger RNA. Then it has to be transported, the messenger RNA has to be transported to the cytoplasm where ribosomes will bind and translate the messenger RNA into protein. Because of this process and transportation of the messenger RNA, we also have to make sure that messenger RNA is protected and not degraded before it's translated. To do this, we actually produced pre-mRNA first in the nucleus, and there's going to be a few steps required, known as mRNA processing, before we form our final messenger RNA molecule that's transported to the cytoplasm. We're going to see that eukaryotes also use 3 different types of RNA polymerases that transcribe different types of genes. And finally, in eukaryotes, the RNA, the messenger RNA, is usually monogenic, which means that a single messenger RNA molecule usually encodes just a single protein. In contrast, we already saw this earlier, but in prokaryotes like bacteria, there's no nucleus, so transcription and translation can occur simultaneously. Their messenger RNA is degraded much more rapidly because it's not protected, like we're going to see soon. For eukaryotes, it is protected. And in prokaryotes, their messenger RNA is usually polygenic, where you'll see on a single messenger RNA molecule, if this is a single mRNA, you might see several genes, like gene 1, gene 2, and gene 3, all on a single messenger RNA molecule. Eukaryotic transcription is much more complex, and in fact, in eukaryotes, we have 3 different types of polymerases, depending on the kind of gene you're going to be transcribing. RNA polymerase will be transcribing ribosomal RNA genes. RNA polymerase number 2 will be transcribing our protein coding genes, and this is the one we're going to mainly be focusing on for transcription. And finally, RNA polymerase 3 transcribes ribosomal RNA, transfer RNA, and small nuclear RNA genes. And in addition to RNA polymerase, it's not shown in this slide, but each type of RNA polymerase in eukaryotes also requires transcription factors. Transcription factors in order to bind to the DNA template strand. Sometimes we abbreviate these TF for transcription factors. If we look at the first step of transcription, or initiation in eukaryotes, the promoter in eukaryotes is much larger and more complex than prokaryotic promoters that we saw earlier. But there is a sequence that we saw earlier in prokaryotes. It was around -10 nucleotides upstream from the initiation start site. In eukaryotes, it's located around the -25 to -35 sequence or bases upstream of the initiation site, and that's known as the TATA box. This is our consensus sequence in eukaryotes, and the sequence is TA, TA, AA. We see the sequence in pretty much all eukaryotic cells, and that's always located within the promoter. This sequence is what our transcription factor recognizes and binds to. And this is the first transcription factor that's laid down. The transcription factor, you can see, includes a subunit over here called TBP, and that stands for TATA binding protein. Binding protein. You don't have to know the small numbers or letters here, but know that once those come down and bind to the TATA box, more transcription factors are recruited until ultimately RNA polymerase 2 is recruited and binds to the promoter near the region of the TATA box. So here, the entire complex with those transcription factors and RNA polymerase II forms our transcription initiation complex. And remember, RNA polymerase II is the type of polymerase. We had 3 different types. This is the type that transcribes protein coding genes on the DNA. So right under this image in our OpenStax textbook, there's a question where they ask a scientist splices a eukaryotic promoter, this promoter, in front of a bacterial gene. So let's pretend there's a bacterial gene around here, and inserts this whole thing, the promoter and the bacterial gene, into a bacterial chromosome. The question is, would you expect the bacteria to transcribe the gene? So if I had a eukaryotic promoter in front of a prokaryotic or bacterial gene, would the DNA be read and form messenger RNA? Would I be able to transcribe the gene? And the answer would be no, it would not work, because eukaryotic promoters will not be recognized by RNA polymerase of prokaryotes. Prokaryotes use different promoters than eukaryotes. So this will not work if you try to mix a eukaryotic promoter with a prokaryotic gene. For a second step of transcription, elongation, there are also differences compared to prokaryotes. In eukaryotes, it's similar to prokaryotes in that messenger RNA is synthesized in the direction of 5 to 3 prime, but this is going to happen in the nucleus of eukaryotes, whereas in prokaryotes it's happening in the cytoplasm since they don't have a nucleus. Additionally, because we wrap our DNA around histone proteins, and it's pretty tightly bound and inaccessible, we actually have to make sure we loosen the DNA, move it kind of away from the histone so that it can be accessed by RNA polymerase during transcription, and then we have to rewind it, retighten it around the histone proteins after transcription is over. And this is facilitated by FACT proteins, a FACT complex. And that stands for facilitates chromatin transcription. That's the protein complex name. So it basically moves along the chromatin and loosens, again, unwraps the DNA, loosens it so it moves away from the histone proteins, and then as you're done with a certain region, it retightens. It reforms that nucleosome complex. Finally, the last step of transcription, our termination step, is different in eukaryotes because for RNA polymerase II, which transcribes our protein coding genes, we don't have what we saw in prokaryotes. There's no row-dependent or row-independent termination. Instead, RNA polymerase II is actually going to transcribe thousands of nucleotides after the gene template. So it just keeps transcribing, and ultimately these nucleotides are going to be removed when we're processing our messenger RNA, when we're turning it from pre-messenger RNA into our final messenger RNA molecule. If I look at RNA polymerase I, there are also differences in termination. RNA polymerase I will recognize or require, a termination protein that's going to recognize a specific sequence to stop transcription. And finally, RNA polymerase III is going to form a hairpin complex that's similar to what we saw in prokaryotes. We're going to focus mainly on RNA polymerase II again for our class. One of the key differences between prokaryotes and eukaryotes is that in prokaryotes, when you make your messenger RNA, it's ready to go and be translated right away. In eukaryotes, however, the pre-messenger RNA that's formed during transcription is not ready to be translated yet. Instead, it has to go through processing before you form your messenger RNA that's ready for translation. So let's look at the 3 things that have to be done during this processing. The first step is something known as Intron splicing. We're going to have to cut out or remove sequences known as introns. These are intervening non-coding sequences that we get that are going to get cut out. The second thing we're going to do is add a 5' methylguanosine cap, and that's sometimes called a G cap. We'll see that at the 5' end of the messenger RNA. So that's usually depicted on the left-hand side of our messenger RNA molecule. The final thing we're going to do is add a 3' poly A tail on the 3' end again of the messenger RNA. So you'll see a bunch of adenine nucleotides. Having the 5' G cap and that 3' poly A tail actually increases the half-life of the eukaryotic mRNA. So it doesn't get degraded as quickly as it does in prokaryotes. And in fact, while in eukaryotes, the messenger RNA can last for hours, so translation can keep occurring. In prokaryotes like E. coli, the messenger RNA only lasts for a few seconds, like maybe 5 seconds or less. So here's another look at that RNA processing. We start with our pre-messenger RNA, and remember in eukaryotes we have to go through extensive processing. The first thing we're going to do is cut out or remove the introns. So the introns are intervening sequences that are not coding. They're non-coding. So those are going to be cut out and removed. We're going to keep regions called the exons. So exons are called exons because they are expressed. These are the important regions of messenger RNA. Then again, we're going to add our 5' G cap and our poly A tail on the 3' end, and that's going to allow the messenger RNA to be stable and not be degraded by different enzymes found within the cell. So here is my final messenger RNA molecule that's ready for translation. You can see the exons have been retained, the introns have been removed, and I can see that 5' end and 3' end that have been protected so enzymes don't degrade the messenger RNA molecule. Notice how small the final messenger RNA product is now that you've cut out all of those introns, those intervening sequences. So here's another look at the same thing, except we're going to focus on the vocab terms. We have exons, which are protein coding sequences that are expressed, and we're going to keep those in our messenger RNA molecule. So this is really our pre-mRNA. And then what we're going to get rid of are the introns. The introns are the intervening sequences that must be removed. We do this through a process known as splicing. So splicing will allow us to remove introns and reconnect the exons together. Notice that we have our 5' G cap over here and our poly A tail over here. And we're not going to look at this yet, but in a later chapter, we'll see that there are untranslated regions on both ends of the mRNA as well. There's a 5' untranslated region and a 3' untranslated region. It'll be important in our next chapter. So splicing is that removal of those introns from our pre-mRNA transcript, and it's catalyzed by these large protein complexes known as spliceosomes. Spliceosomes over here. These spliceosomes are made up of proteins in combination with small nuclear RNA molecules, so snRNAs, and this one there's a typo there, so this should say snRNAs. And I can see that the spliceosome, which is this whole complex, is going to bind to and get rid of my intron, which is in green right here. Although the intron is non-coding, it's an intervening non-coding sequence, there are marks at the ends of the introns. So the 5' end of the intron has a G and U nucleotides, and the 3' end of these introns have A and G at their end. And spliceosomes will recognize these two sequences, bind to that region to remove the intron over there. Here's another look at the same thing. In this image, the whole thing over here is the spliceosome, and we can see these small molecules are those small nuclear RNAs. This silver segment right here is the intron that we're going to splice out, and these would be, over here would be the 5' and 3' end of the introns that are recognized by the spliceosome. If we have mutations in the DNA, which will lead to mutations in the messenger RNA, that could lead to splicing errors. So if these sequences were lost, these recognition sequences for the spliceosome were lost, then we might not be able to cut out the intron, and that could lead to a protein that ultimately becomes non-functional. Or if there are mutations in the small nuclear RNA sequences that are part of the spliceosome, that could also cause problems with cutting out introns and producing normal proteins after you translate the messenger RNA. Just as you did for the previous two videos, after you finish watching this third video, on our Canvas website, you'll see an external link that'll take you to this animation page. I'd like you to click through the animation. It's very short. It reviews the process of RNA splicing and the processing of pre-mRNA. So our pre-messenger RNA will be processed, converted to messenger RNA, and then translated. But ribosomal RNAs and transfer RNAs are not going to be translated. They stay in their RNA state. Pre-ribosomal RNAs will be transcribed and processed, and they're going to be assembled into the ribosomes within the nucleolus. Remember, this structure is found within the nucleus. Pre-transfer RNAs are going to be transcribed and processed in the nucleus. Then they're going to be shuttled to the cytoplasm, and they're going to be bound to free amino acids to help with protein synthesis. So this is a picture from our textbook that shows us the image or the structure of transfer RNA. And the function of transfer RNA is to bring amino acids to the growing polypeptide chain during translation. So we'll see these in the next video. They're going to be interacting with ribosomes to help grow our polypeptide chain. For the tRNA, there are two important sites. One is the amino acid attachment site, and in this case it happens to be the amino acid phenylalanine. On the other side, we have an anticodon that will base pair or hydrogen bond with the codon that are found, or the codons that are found on the messenger RNA. So if this is the anticodon, then the codon would be UUC, and this is what encodes phenylalanine. So if we go back and look at that codon chart, the genetic code, you'll see that UUC on the messenger RNA encodes phenylalanine for its amino acid. And this is from a different textbook, but I wanted to show you other images or depictions of transfer RNA. Sometimes you'll see something that looks more like this. This is where the amino acid would be bound, and over here is where the anticodon is located. Or sometimes you'll see something like this. Again, this is where amino acid would be bound, and here's my anticodon. If the anticodon is ACC, what do you think the amino acid is? So I would give you the codon chart. Let's see in the next slide. So here is my codon chart. Again, this is the genetic code. If this is my anticodon, then what that means is I know my messenger RNA, where the codons are housed, would be complementary. So that would be A pairs with U, that would be GG. And if I look at my codon chart, UGG, UGG, encodes the amino acid tryptophan. So that's the amino acid that would be attached on the other side of the transfer RNA molecule. So notice that the amino acid is determined by the codon, not the anticodon. So we look at the codon chart here. All right, and that takes us to the end of our third video. In our fourth and final video for Chapter 15, we're going to be looking at the details of translation. How do we go from messenger RNA to our final polypeptide or protein product?